So now let me discuss the classification of the receptors. If you see these receptors, remember the receptors are classified into 4 types, right? And what is the basis for their classification? Remember, based on signal transduction mechanisms, the receptors, they are classified into 4 types. Alright. So if you see the classification of the receptors, like they are of 4 types. Alright, they are of 4 types. And what is the basis for the classification? Based on signal transduction mechanisms. Alright, so based on signal transduction mechanism, they are classified into 4 types. Now what are those 4 types? Let me discuss this. One, they are called as G protein coupled receptors, right? First one, it is called as G protein coupled receptors. Alright. Now this G protein coupled receptors, they are also called as metabotrophic receptors, right? They are also called as metabotrophic receptors. That is your G protein coupled receptors. So the second type are the inotropic receptors, right? The second type are the inotropic receptors. Next, after this inotropic receptors, the third variety of, or the third type of receptors is your enzymatic receptors. Alright, it is your enzymatic receptors. And the last 4th one will be your intracellular receptors, right? Intracellular receptors. So these are like the classification of the receptors based on the signal transduction mechanisms. Now what I will do is, first let me discuss about the G protein coupled receptors or metabotropic receptors. Now, so if you see the discussion on G protein coupled receptors, alright, so this G protein coupled receptors, they are also called as the metabotropic receptors. Now first and foremost, very important point what you should remember here is, this G protein coupled receptors, these are heptahelical receptors, right? First very important point, these are heptahelical receptors, right? These are heptahelical receptors, which are also called as the serpentine receptors. Right, which are also called as the serpentine receptors. Now what do you mean by this word, the heptahelical? That means the word hepta, it stands for the 7, the number 7. Heptahelical means, that is, they have 7 transmembrane spanning segments, right? So they have 7 transmembrane spanning segments. Alright, so that is what is the meaning of the heptahelical receptors. Now let me discuss this G protein coupled receptors. Now what these receptors do? Remember, the drugs, they come and bind to the receptor, right? The drugs, once they come and bind to the receptors, which in turn activates the G protein, right? So what will happen is, the drugs, they come and bind with the receptor, right? They come and bind with the receptor. Now once the drug bind to the receptor, now the receptor will in turn activates the G protein. Okay, so this will further activate the G protein. Alright, this further activates the G protein. Okay, so now once this G protein is activated by the receptor, wherein the receptor is bound by the drug, now this is called as GTP activated protein. Right, this is called as the GTP activated protein. Alright, now you take this G protein. So ultimately what is happening here? The drug is coming and binding with the receptor. Now this in turn it activates the G protein, right, which is nothing but GTP activated protein. Now you take this G protein. Remember, this particular G protein, it has 3 subunits. Right, G protein, it has 3 subunits. Now what are those 3 subunits of the G protein? It contains the 3 subunits like alpha, beta and gamma. So these are the 3 subunits of the G protein. Now remember, when all 3 are joined together, right, then that particular G protein is inactive. Right, when all the 3, that is alpha, beta, gamma, all the subunits, when they are joined together and that particular G protein is inactive. Right, now all these are joined together along with GDP, that is guanosine diphosphate. Okay, so when all the 3, right, when all the 3 are joined together along with GDP, right, when all the 3 are joined together along with the GDP, and that particular G protein, right, that particular G protein is inactive. Right, that particular G protein is inactive. Now the question is, when does this particular G protein gets activated? So let me shortly revise the part what we have discussed until now before moving on to the further part of the discussion. You see here, we are discussing the classification of receptors. Now these receptors, they are of totally 4 types based on the signal transduction mechanism. They include the G protein coupled receptor, which are also called metabotropic receptors, inotropic receptors, enzymatic receptors and intracellular receptors. So these are the 4 types of the receptors. Now what we are discussing is mainly about the G protein coupled receptors. Remember, these are heptahelical receptors, which are also called serpentine receptors. And why are they called heptahelical? Because they have 7 transmembrane spanning segments. And now if you take this particular receptors, the drug, they come and bind to the receptors, which in turn activates the G protein. Right, and that is nothing but your GTP activated protein. Now where is this particular G protein present? This G protein, it is present within the cell. Now once the drug comes and binds with the G protein receptor, then that in turn activates the G protein which is present within the cell. Now this particular G protein, it consists of 3 subunits, alpha, beta and gamma. When all 3 are joined together, then the G protein is inactive. All these 3 are joined together along with GDP. Now let me tell you, when does this particular inactive G protein gets activated? So now let me tell you, when does this gets activated? So remember, when GTP, now what is there in the inactive component? GDP is there, guanosine diphosphate. Instead of that, if GTP, that is guanosine triphosphate, so when GTP replaces GDP, then the alpha subunit separates from beta and as well as the gamma subunit, and then it becomes activated, right, and then it becomes activated. So remember, when GTP replaces, right, when GTP replaces GDP, then what will happen is the alpha subunit, right, the alpha subunit, separates from, right, alpha subunit separates from beta and as well as the gamma subunit, right, separates from beta and as well as gamma subunit, and it becomes activated, right, it becomes activated. So now what is activated now? The G protein coupled receptor is being activated, right? How? When the alpha subunit separates from the beta and as well as the gamma subunit, and when GDP is replaced by GTP, GDP stands for guanosine diphosphate, GTP stands for guanosine triphosphate. So then this particular receptor gets activated. Now let me tell you, like, actually what is the thing what we are discussing now. Remember, you take the drugs in the pharmacology. The drugs in the pharmacology, they don't act directly and you have an effect. The drugs, they have to act through various receptors, right? The drugs, they have to act through various receptors for their action. So what are we discussing now? We are discussing what are the various types of receptors through which the drugs will be acting, right? What we are trying to discuss is what are the various receptors through which the drug will be acting, and what we are trying to discuss in depth is what is the mechanism of activation of that particular receptor, right? And once that receptor is activated, then only the drug will show its pharmacological action. So that is what we are trying to discuss now. All right. So now once this particular G protein coupled receptor is activated, now activated alpha subunit. Now what is there in the activated G protein coupled receptor? Only alpha subunit is there. Activated alpha subunit may result in one of the three actions. Okay. So alpha subunit separates from beta and gamma subunit and it becomes activated. Now this particular activated alpha subunit may result in one of three actions. Right, activated alpha subunit will result in one of the three actions. Now let me tell you what are those three actions. Number one, first one is either activation or inhibition of the enzyme adenylyl cyclase. Okay, once the alpha subunit is activated, there will be one of the three actions. I am telling you what are those three actions. Number one, right, number one, activation, that is GS, right, that is G protein coupled receptor stimulated. Activation or inhibition, right, activation or inhibition. Inhibition is done by GI. GI stands for inhibition. So activation or inhibition of enzyme, right, of enzyme adenyl cyclase, right, activation or inhibition of the enzyme adenyl cyclase. Now what is the second action? Second action is that activation of phospholipase C. Activation of phospholipase C, and this particular activation is done by GQ, right, this particular activation is done by GQ. Now you take the third action. The third one is stimulation or inhibition of the ion channels. Okay, stimulation or inhibition of ion channels. So that is the third action. So remember, once the alpha subunit is activated, one of the three actions will occur, right? Either there will be activation or inhibition of the enzyme adenylyl cyclase, either there will be activation of phospholipase C, or there will be stimulation or inhibition of the ion channels. Now let me discuss one by one again now. Now remember, once there is activation of the adenylyl cyclase, right, what will happen? Remember, like we have a second messenger which is called as the cyclic AMP. So once there is activation of the adenylyl cyclase, it changes the concentration of the cyclic AMP, right? So this will change the concentration of the cyclic AMP. Now, what do you mean by the word second messenger? Remember, once the receptor is activated, the activated receptor will show the pharmacological action of the drug through that particular second messenger, right? And what is the second messenger? Once there is activation of the adenylyl cyclase, it is your cyclic AMP. So remember, once there is activation or inhibition of the enzyme adenylyl cyclase, it changes the concentration of the cyclic AMP, right? And this particular cyclic AMP, it acts by activating the protein kinase, okay? So this cyclic AMP, it acts by activating the protein kinase, right? It acts by activating the protein, that is protein kinase. Alright, now let me tell you the examples of the receptors which act by the activation or the inhibition of the G protein. Okay, the examples if you see here, right, if you take the examples of the activation or the inhibition of the enzyme adenylyl cyclase, these particular examples, they include beta receptors, right? What are your beta receptors? Remember, we have, like in the further part of the discussion, like we will be discussing that there is a system which is called as adrenergic nervous system. Adrenergic nervous system, the neurotransmitter is epinephrine and as well as norepinephrine. Epinephrine and norepinephrine, it acts via the receptors alpha and beta receptors. And if you take the beta receptors are the one which acts by the activation of the adenylyl cyclase enzyme. So what the beta receptors will do is, beta receptors will increase the cyclic AMP, right? Beta receptors will increase the cyclic AMP. Now we have one more receptors which are acting via the activation or inhibition of the enzyme adenylyl cyclase, and that is nothing but your somatostatin, right? The other one is somatostatin. Alright, now if you take this particular somatostatin, it works by decreasing the cyclic AMP. Right, it works by decreasing the cyclic AMP levels. Is that clear? Now, so that is about your first one, activation or inhibition of the enzyme adenylyl cyclase. Now we will move on to the second, that is activation of the phospholipase C by GQ. Now, what this particular enzyme will do? Now, what was your adenylyl cyclase doing? Adenylyl cyclase was changing the concentration of the cyclic AMP, and this will further activate the protein, that is protein kinase. Now, what will happen once there is activation of the phospholipase C? Remember, this particular enzyme converts PIP2 to IP3 and DAG. Okay, this enzyme, right? This enzyme, it converts PIP2, right, IP3 and as well as DAG, right, IP3 and DAG. Now you take PIP2, this stands for phosphatidyl inosyl phosphate 2, and this will be converted into inosyl triphosphate and as well as diacyl glycerol. Now you should remember what are the receptors, what are the examples of the receptors which will act via the activation of the phospholipase C. Remember, the receptors which act through this particular activation of the phospholipase C is your alpha receptors and vasopressin receptors. Okay, so the examples are alpha receptors and as well as V1 receptors, which are nothing but vasopressin receptors. Vasopressin, it acts via three receptors, V1, V2 and V3, whereas V1 receptors, they are acting via the activation of phospholipase C. Now, what will happen once there is activation of the phospholipase C? The final result is increase in the intracellular calcium. Okay, so once there is formation of IP3 and DAG, there is increase in the intracellular calcium. Right, there is increase in the intracellular calcium. So that is what is your activation of the phospholipase C. Next, you take the third one, that is stimulation or the inhibition of the ion channels. Remember, what are the receptors which act via stimulation or the inhibition of the ion channels is the example, right, example, the M2 receptors, right, M2 receptors of the acetylcholine. Right, M2 receptors of the acetylcholine, they act via the stimulation or the inhibition of the ion channels. Alright, now out of this, remember, let me tell you which are the second messengers and which are the third messengers. Now remember, you take the second messengers. Right, which are the second messengers? The second messengers, they include, right, they include cyclic AMP, they include inosyl triphosphate. So the second messengers are the cyclic AMP, inosyl triphosphate and diacylglycerol. And if you take the third messenger, right, so the third messenger is the calcium. Right, the third messenger is the calcium. Alright, now, so what do you mean by this second and third messenger? Remember, this particular drug, once they come and bind with the receptor, directly it will not show the pharmacological action. The drug should come and bind with the receptor activates either of these. Now this will send the information to the nuclei via these particular second messengers and as well as the third messengers. Alright, now remember, after the action, right, after the action, the intrinsic GTPase activity of the alpha subunit results in joining it with beta and as well as gamma subunits, and thus G protein is available for action again. So what I am trying to tell you here is, now the alpha subunit, once it is activated, it is doing either of these three actions. Right, after the actions, by activated alpha receptor, the intrinsic GTPase activity of the alpha subunit, what it will do is, after activating all these three, the alpha subunit, it will again rejoin with the beta and as well as the gamma subunit, and thus again the entire G protein is available for action once again. Right, so that is what you need to remember here. So let me shortly revise about the G protein coupled receptor. So in the classification of the receptors, I have said you that we are having four types. One is G protein coupled receptor, right? Second is your enzymatic receptor. The third is your inotropic receptor, and the fourth is your intracellular receptors. Now if you take this particular G protein coupled receptors, they are metabotropic receptors, and these are heptahelical receptors, that is they have 7 transmembrane spanning segments, that is they are also called serpentine receptors, and the drugs, they bind to the receptor, which in turn activates the G protein, right, which is nothing but GTP activated protein. Now the G protein, it consists of 3 subunits, alpha, beta and as well as gamma. When all 3 are joined together along with GDP, then the G protein is inactive. Now when GTP replaces the GDP, then the alpha subunit separates from beta and gamma and it becomes activated. Now, the activated alpha subunit may result in one of the three actions, that is activation or inhibition of the enzyme adenyl cyclase, activation of the phospholipase C, stimulation or inhibition of the ion channels. Now, you see the activation or inhibition of the enzyme adenyl cyclase. It changes the concentration of the cyclic AMP that acts by activating the protein kinase. Now, if you take the examples of these particular receptors, these include beta receptors and as well as the somatostatin receptors. The beta receptors, they will increase the cyclic AMP, somatostatin, they will decrease the cyclic AMP. Now you take the second thing, the activated alpha subunit may also cause the activation of the phospholipase C. Now this activated phospholipase C, this enzyme converts PIP2 to IP3 and as well as DAG. The final result is increase in the intracellular calcium, and thus the action of this particular activated phospholipase C is on the alpha receptors and as well as the V1 receptors, which are nothing but vasopressin receptors. And the third one is stimulation or the inhibition of the ion channels, that is the example is the muscarinic receptors, the M2 receptors of the acetylcholine. Now you take the second messengers here that include cyclic AMP, inosine triphosphate and diacyl glycerol, they act as the second messengers, whereas you take the calcium, it acts as the third messenger. Now the activated alpha subunit, after activating these three enzymes, this activated alpha subunit will again go and bind with the beta and gamma subunits, and thus the G protein is available for the action once again, right? So this is about the discussion about your G protein coupled receptors. Now after having discussed about the G protein coupled receptors, now let me discuss about the inotropic receptors. Right, let me discuss about the inotropic receptors. Now, so if you take this particular inotropic receptors, remember what will happen here. In inotropic receptors, the drug, it comes and binds directly to the receptors, which is located on the ion channel. And this particular inotropic receptors, they do not require the mediation by the G proteins, whereas G protein coupled receptors, they were mediated by or via the G proteins, whereas here for the activation of the inotropic receptors, there is no requirement of the G proteins. Okay, so here what will happen is the drug binds directly to the receptor. Alright, the drugs bind directly to the receptor. And now where is this receptor located? Remember, this particular receptor, it is located on an ion channel. Okay, receptor located on an ion channel. Okay, receptor is located on an ion channel. Now a point is, this inotropic receptors, they do not require the mediation by G proteins, right? Requirement of G proteins is not there for this particular inotropic receptors. Now a very important multiple choice question regarding the inotropic receptors is, remember, the inotropic receptors are the fastest acting receptors. Alright, these are the fastest acting receptors. Okay, so this is one of the very important point. Now let me tell you the examples under this particular inotropic receptors. Now what are the examples of this inotropic receptors is, number one, you have gamma amino butyric acid A, that is GABA A, and the other one is the nicotinic receptors, that is we have NM and as well as NN. So NM and as well as NN, right, they are also the ionotropic receptors, which are nothing but nicotinic receptors. Then we have NMDA receptors, right, then we have NMDA receptors. So NMDA receptors, these are the receptors of glutamate, right, NMDA receptors, these are the receptors of the glutamate. Alright, next, the other receptors, other ionotropic receptors are, they are 5-HT3 receptors. Alright, they are 5-HT3 receptors. So these are all your ionotropic receptors. Okay, now after having discussed about the ionotropic receptors, now let me discuss about the enzymatic receptors. Right, let me discuss about the enzymatic receptors. Now remember, this type of receptor, it has two sites. One is the intracellular site and the other one is the extracellular site. Okay, so this type of receptor, it has two sites. Alright, it has two sites, right. The drug, it binds on the extracellular site and as well as on the intracellular site. Okay, so it has two sites. One is the extracellular site and the other one is the intracellular site, right, the extracellular site and as well as the intracellular site. The drug, if you take, the drug binds on the extracellular site. Alright, the drug binds on the extracellular site. And if you take the intracellular site, the intracellular site, it has enzymatic activity. Right, it has the enzymatic activity. And what is that particular enzymatic activity? Mostly tyrosine kinase activity. Right, mostly tyrosine kinase activity. Alright, so this is your enzymatic activity, which is the intracellular. Now remember, this enzyme can be activated by a pathway which is called as the JAK-STAT pathway. Alright, so this tyrosine kinase enzyme, this enzyme can be activated by, right, it is activated via JAK-STAT pathway. Alright, it is activated via the JAK-STAT pathway. So that is your enzymatic receptors. Now after this, let me discuss about the intracellular receptors. Right, let me discuss about the intracellular receptors. Now these types of receptors, they are slow acting. Right, these type of receptors, they are slow acting or the slowest acting. Now where are these present? Remember, this particular intracellular receptors, they are either present on the, right, either they are present in the cytoplasm, right, or this particular intracellular receptors, they are present in the nucleus. Right, they are present either in the cytoplasm or within the nucleus. Now if you take the intracellular receptors which are present within the cytoplasm, now intracellular receptors which are present in the cytoplasm, they include the receptors for the glucocorticoids, right, they include the receptors for the glucocorticoids, they include the receptors for the mineralocorticoids, right, they are also present within the cytoplasm. Next, they include the receptors for the vitamin D, right? They include the receptors for the vitamin D. Now, if you take the receptors which are present within the nucleus, the receptors which are present within the nucleus, they include the receptors for the thyroid hormone, that is T3 and as well as T4, and then we have the receptors for the retinoic acid, right? The receptors for the retinoic acid. Then we have the receptors for estrogen. Then we have the receptors for the progesterone. Alright. Then we have the receptors for the testosterone. Right. Then we have the receptors for the testosterone. Alright. Now, remember both type, right? Both types of receptors, that is cytoplasmic receptors and as well as the nuclear receptors. Both type of receptors, finally they act by the nuclear mechanism, right? That is by affecting the transcription. Okay, so either you take the cytoplasmic receptors or the nuclear receptors, they act by the nuclear mechanism. Right, they act by nuclear mechanism. What is that nuclear mechanism? That is by affecting transcription. Right, that is by affecting transcription. So that is your intracellular receptors, right? Let me shortly revise all the receptors now. So we have already discussed the G protein coupled receptors. Now let me revise about the ionotropic receptors. If you take this particular ionotropic receptors, remember the drugs, they directly bind to the receptors located on an ion channel without mediation by G proteins, and these are the fastest acting receptors. And what does they include? It includes GABA-A, nicotinic receptors, that is NM and NN, and then we have NMDA receptors, that is the receptors for the glutamate, and as well as the 5-HT3 receptors, that is 5-hydroxytryptamine. Next, you take the enzymatic receptors. This type of receptors, it has two sites. The drug binds on the extracellular site and as well as the intracellular site, right? So on the extracellular site, it is required for binding of the drug, whereas the intracellular site, it has the enzymatic activity and mostly tyrosine kinase activity, and this enzyme can be activated via what is called as the JAK-STAT pathway. Now you take the intracellular receptors. Remember the intracellular receptors, these type of receptors, they are slow acting, and they may be, the receptors may be either in the cytoplasm or within the nucleus. You take the cytoplasmic receptors, they are mainly for your glucocorticoids, mineralocorticoids, vitamin D, and the nuclear receptors, they are mainly for your thyroid hormone T3 and T4, retinoic acid, estrogen, progesterone, and as well as testosterone. And both types of receptors, finally they act by the nuclear mechanism, that is by affecting the transcription. Now let me tell you the examples of the enzymatic receptors. Now if you take the examples of the enzymatic receptors, remember what are the hormones which will act via the enzymatic receptors? They include insulin, right? They include growth hormone, right? They include growth hormone, and the other hormone, that is prolactin, right? Other hormone, that is prolactin, and the other hormone, that is cytokines, right? Other substances, that is cytokines. So remember, insulin, growth hormone, prolactin, and cytokines, they act via the enzymatic receptors, right? This is one of the very important multiple choice question here.